company located in Austin, Texas. It was born up in the Bay Area and moved it down there in 2010. I'm actually standing in for my Chief Scientific Officer, Steve Pastrelski, who some of you may have worked with during his days at Amgen, Alza, PowderJect. I don't know where Chris and he worked together. He spent most of his career in peptide protein formulation. And is most recently the head of VP of R&D at Pharma R&D at Amylin Pharmaceuticals and ran the Bidurian program for them. Thank you, Chris, for inviting me. What Xeris is working on, or special sauce, I guess, if you will, is that most biologicals are very unstable in water. They start their lives out as a lyophilization solution, like up here with a lot of excipients in it, primarily in there to enable the reconstitution phase. What we've done is figured out a way to dramatically simplify the powders, the lyophilization powders, and then mix them with a FDA-approved non-aqueous solvent. But the point is that as they're either stored as dry powders and have to be reconstituted, or stored as aqueous solutions and stored in a refrigerator. Insulin probably being the best injection or best example. Painful injections, high volumes, complicated delivery, and it's designed for water-based delivery. And so what we've done is eliminated the water, like I said, reduced the complexity of the lyophilization solutions, and make powders, much lower volume powders, mix them with FDA-approved solvents or diluents. And what this allows us to do is, at the time of manufacture, load them into auto-injectors, multi-dose syringes, or in the case of glucagon, which I'm going to talk about, you can put it into pumps. They're room temperature stable, room temperature stable, so it allows people that usually have to mix them up as kits to walk around with these kind of products in their briefcases, backpacks, whatever. So what it allows for is dramatically lower injection volumes, very room temperature stable, easy to administer, and more personalized dosing. So we have two platforms, Xerisol. It's for non-aqueous solution, primarily for delivery of peptides, and recently we've discovered it's very applicable to small molecules. You get 5 to 50x reduction in injection volumes, and it's a faster to market opportunity because of you're able to use on the market auto-injector pens like I have with me today that we're using for our glucagon program. Our lead program is glucagon. Has the opposite effect of insulin, increases blood glucose. And then we have our Xeriject platform. It's non-aqueous suspension paste. It's primarily focused on delivery of large biologics like monoclonal antibodies. We're working on one right now. And you can get high concentrations of the injection. You can get take IV solutions down to a subcutaneous injection, which, as many of you know, is where the monoclonal antibody space is going, even in the oncology products, because of the cost of hanging an IV bag on someone. So already talked about it. The big opportunity here is for delivery of monoclonal antibodies and large proteins. So just an important point here is all the non-aqueous solvents we're using are FDA approved for use in other parenteral products. So Xerisol summary: non-aqueous solutions of peptides, biocompatible solvents, polar A products. Best example is DMSO, which we're using in our glucagon program. A lot of our technology and know-how is in a proprietary formulation process. So you get significantly increased solubility and stability. And there is also, because the significant reduction in injection volumes and some of the solvents we're using, like DMSO, there's a potential for reduced injection site reactions. So G-Pen, glucagon pen, is our lead product. Just a quick target profile on it. It's for emergency treatment of severe hypoglycemia and insulin use, all diabetics actually. Best-in-class performance. There's no reconstitution. The current product is a kit with a 1 mg of glucagon in a sealed vial that you have to reconstitute with 1 ml of water. It's an SC injection, and it's a 200 microliter injection volume down from the 1 milliliter that's in the Lilly and Novo kits. Two-step auto-injector. You simply pull off the end and inject your child. And most severe hypoglycemic events happen between 11 p.m. and 2 a.m. So it's parents treating their children or treating their loved ones, or if they don't get treated, some actually self-treat. About 11% we found in a big survey self-treat themselves, believe it or not. But don't get treated. It's you get seizures, coma, and there's a phrase in the industry called dead in bed, unfortunately. It's a 200 microliter injection, 1 second hold down time, and we've got data now showing that we can get 2 years of room temperature stability. Go. Here's some early PK/PD data. Let's see. Is this got a... So glucagon and NMP, glucagon and DMSO, glucagon NEAT and DMSO, and then aqueous reconstituted glucagon. You can see from the various curves here is that they produce what are statistically equivalent PK curves, and same situation here over with blood glucose levels. You're seeing peaks in about 15 minutes with all of them. So that's about all I got to say about that. Development status, like I said, we're darn near in the clinic with this. We've had our IND-enabling program agreed upon with the FDA in terms of our clinical program, preclinical talks. The lead clinical formulation is chosen, and we've scaled the clinical volumes. Tox engineering batch is manufactured, released, and our clinical batch has been manufactured, 2,000 pre-fill 1 ml syringes. We've done our bridging farm tox study is completed in the rat, and we haven't got our PK data, but there's no systemic or injection site related issues. So we're getting ready to go into the clinic down at the Texas Diabetes Institute in San Antonio, Texas. I'm actually a co-PI on this. If there's any NIH folks in the room here, this is an SBIR, a $1.1 million SBIR grant from the National Institute of Diabetes, Digestive, and Diseases of the Kidney Diseases. So we're proud to be working with NIH and Dr. Ralph DeFranzo on this. This is actually a suite of products, if you will, or a portfolio. The first one is a glucagon rescue pen, and it's a 5 milligram per ml solution, and we're leveraging it across a mini dose pen. And sorry, folks, go back here. This is a glucagon mini dose pen is a microdosing of glucagon. It's a non-caloric treatment for mild hypoglycemia. And there's a strong market potential we've already tested through Diabetes Q&A in San Francisco. It's about a 5,000 patient cohort that we've tested with. And then finally, the artificial pancreas. We just won a $1 million grant through NIDDK. We're co-PIing with Dr. Ken Ward at Oregon Health Science University. This will put our 5 milligrams per ml solution in a pump in diabetic patients in sometime in late 2013 up at the Oregon Health Science University Hospital. We're also teaming with Ed Damiano at Boston U, who is one of the other leading artificial pancreas or bionic pancreas guys. A second product we're working on is Xeris diazepam for epileptic seizures. This is actually funded by the Epilepsy Therapy Project. It's a highly concentrated solution of diazepam. And again, we'll put it into a single-use auto-injector pen like this Molly pen here. Highly stable. This just And the terrific thing about this, an aqueous solution, to get the right dose, you have to go up to 3 ml injection that nobody will sit still for, stabbed into their legs. So multiple solvent systems, where we are with this is we're in a rat PK study with screening several formulations right now. Okay, Xeris formulation platform, just talked about this briefly. It's for highly concentrated suspension of dry powders, actually pastes. They're sort of thixotropic pastes, solids when no pressure on them, and then they flow like liquids when you put pressure on them. So 2 categories, highly potent drugs up to 3 milligrams, drug placed in an aluminum syringe, and then large doses up to 500 milligrams per mil. And this is where you come into play on large molecules like monoclonal antibodies for SC injection. We've actually done some initial proof of concept testing. This is a 1.5 inch 25 gauge ultra thin wall needle with a engineered piston in it, and we used it to deliver a Xeris formulation of glucagon in a rat PK study. We've got a filling system we could reproducibly fill and finish this, and then for larger volumes we can do direct transfer from a filling syringe. So everybody has cute names for collaborations. Ours is Xeris Screen. Feasibility project goals would be for a monoclonal antibody maker would be to develop a Xerisol or Xeriject formulation, demonstrate injection delivery of candidate formulations, get into a rat PK study and look at short-term stability biologic, and work with a partner to demonstrate in vivo and preclinical pharmacology. And we're partnered up with some people, but it's under the NDA, so I'm rude to share that information. So IP status, our patent on our Xeriject, which is our base technology. This is the semi-solid formulations, and we're doing additions to this base technology right now with molecules. Glucagon is included in that. Xerisol is this liquid formulation, primarily focused on peptides, small molecule formulations, patent filed. So products, Xerisol glucagon, Xerisol ultra rapid acting insulin, which we're working on, which allows you to deliver, basically we'll be able to deliver room temperature physiological monomeric insulin. It's very stable. Early stages, it's very stable, but it's pretty amazing. It acts like glucagon does. It's steady. Thermodynamic state is in monomeric form. So we're early on that. So thanks very much for your and be gentle on me. I'm a toxicologist by training, but appreciate the opportunity to present today.
Thank you, John, for a very different and honestly very simple approach to the glucagon problem. Does anyone have questions? Walid?
No injection site reaction?
You know, we haven't done big molecules like monoclonal antibody yet, to be determined. But with in our recent PK tox study with glucagon, none. Very clean.
And when you inject it, there is no precipitation after that in the body, or what happened?
Well, I think from the pharmacokinetics we've seen, there isn't any compared to, we ran it against the Lilly formulation. We ran it against the actual Lilly material. And so I'm not sure I can answer that, except that the pharmacokinetics look biosimilar, bioequivalent.
And why you go to glucagon? I want to ask about the insulin, but you apparently...
Good question.
Because insulin market is what glucagon is 1/100th of insulin.
So this is probably, I call it the Rodney Dangerfield of hormones, in the sense that because of the way that it's packaged right now, until we came along with our formulation technology, there wasn't a room temperature stable glucagon solution. And so it ends up in a vial. and it's very unstable. It starts to degrade and fibrillate almost within minutes of mixing it up. So doctors don't like to prescribe this, and patients, caregivers really don't like to use it. It's 9 steps to put it together to actually do an injection. So it's about $150 million market right now. Conservatively, we think we can go to 3.5–400 million with, but we have to realize that our mini dose is probably going to cannibalize a bit of that marketplace, keeping people out of severe hypoglycemia, which is, you're here to take care of patients first, right?
So how far you are with the insulin, monomeric insulin?
Early stage, but we're pretty darn excited about it. Think about it. Walk around room temperature stable monomeric insulin is physiologic. It's kind of disruptive, right? It's a ultra-rapid acting insulin market is around $5 billion right now. So we hope we can get a little bit of that, or our commercial partner can.
But will be quick-acting insulin there, right?
Yes. Yeah. And where the exciting part of that is to put it on board in a bionic pancreas alongside our glucagon. So you've got people, they've got a bionic pancreas. So pretty exciting.
And GLPs?
Sorry?
And GLPs, peptides?
Well, we're doing something. I don't really want to talk about it yet.
Oh, okay.
Yeah. But you're thinking you're going the right spot. So thank you very much.
Yeah, thank you, John. And some of you may know Steve Prostrowski, who I worked with at Amylin, but when I look at this presentation, it looks like a combination of all of the places where he's worked before, which is fine. That's how we do these things. Thank you very much, John.
You're welcome. Appreciate it.